Greetings, it's Kate Scow again, your humble servant, delivering now for Soil Microbiology, Soil Science 111, Lecture 2, Part 2. And we are in a lecture about soil habitat and factors regulating microbial activity. And that's what Part 2 is about. So what we're going to talk about in this lecture is these different physical and chemical factors that have impacts and govern rates at which microorganisms grow and are active in soil. And we're not really going to look at nutrients here, carbon and nitrogen in particular. That will come up later when we look at the biogeochemical cycling. And you also know we already have looked at biological interactions of among different groups of organisms in soil. But we're focusing, as I said, on the physical and chemical. So we're going to look at oxygen, pH, temperature, soil, water, osmotic potential, toxins, and bioavailability, which is an extremely important topic in soil and something that very much influences how fast things happen in soil. So let's just get started. And what I want to do is quickly go through these important factors, and then we will have a class exercise where you will walk through and kind of create sort of a model describing what will determine how fast a microorganism will carry out an activity based on these relationships that we know. In some cases, really clear relationships between these factors and microbial activity. In some cases, it's kind of complex. And in some cases, you think you're looking at one factor like soil water, but manipulating soil water is going to change some other factor in soil that may actually be what is responsible for the change in activity. Okay, let's get going. And let's get going. So oxygen. Oxygen is extremely important. Oxygen is the most favorable electron acceptor that is used by soil organisms. You're going to find the fastest rates of microbial processes associated with organisms that can use oxygen as an electron acceptor. So oxidation reduction reactions are what is involved in this, are what are going to be sensitive to oxygen. And as you recall, redox is a measurement of the oxidation reduction potential of a habitat. And so there are direct effects of oxygen on microbial activity. And in this case, there are particular enzymes that require oxygen, molecular oxygen. Enzymes that have names like oxygenases and oxidases, which are very important enzymes for breaking down complex organic material in soil, as well as other involved in other processes. So they need oxygen, so they will not be active in the absence of them. And also oxygen can be toxic to microorganisms, and so there can be direct toxicity effects. And I already have alluded to this in an earlier lecture where we talked about the ability to withstand an aerobic environment takes heroic activities. So the byproducts that are formed by molecular oxygen interacting with, for example, organic or inorganic compounds are powerful oxidizing agents. And these oxidizing agents include things like peroxides, free radicals, and they can end up impinging on and reacting with cells and causing a lot of damage to cells. So any organism that's able to grow in the presence of oxygen will have to deal with these toxic products. So you have to be ready to withstand this not very pleasant environment in the presence of oxygen. So we're going to look at some different groups of organisms, and you remember these terms: obligate aerobes, facultative anaerobes, obligate anaerobes, and then also you remember the like a microaerophilic organism, an organism that uses only low concentrations of oxygen. So the effect of oxygen on the growth of these organisms: with an obligate aerobe, they're going to have oxygen present to be able to carry out their reactions, and they'll die in the absence of oxygen. And let's group that together with facultative anaerobes. As you recall, if there is oxygen present, they'll use it, but they will also use other compounds as electron acceptors if there is no more oxygen present. So that would be like nitrate and sulfate. So both of these groups of organisms that can function in the presence of oxygen have enzymes that will actually work to detoxify these environments, these toxic chemicals that form in the presence of oxygen. And these enzymes include catalase and superoxide dismutase, which are generated by the organisms and will disable these compounds and detoxify them. If you put peroxide in like your sink and you see bubbling, that's often the result of that, the catalase inside organisms that are aerobes that are growing in like biofilms in your sink, and that bubbling is this CO2 that's formed by this catalase detoxifying the peroxide, forming this gas that you see. It's kind of cool. Okay, and there's some examples of organisms. Wow, they got their names broken up. Anyway, then obligate anaerobes do not require oxygen, and they can only survive in the absence of oxygen. And if oxygen is present, they lack the enzymes that are able to neutralize the oxygen, and they will just drop dead. Clostridium is an example of that. Do you remember Clostridium? Clostridium makes a certain kind of spore. Remember? Makes an endospore, like bacillus. Okay, and then we can go on into more detail. There's microaerophiles, which can live in the presence of oxygen, but it can't be very high. Anyway, those are effects of oxygen on organisms. Now let's talk about pH. So pH has direct effects on microorganisms and indirect effects. Indirect effects are probably more important than direct effects, but direct effects include changes in enzymes that have a pH dependency. If they are requiring alkaline conditions and the pH drops to acidic conditions, these enzymes may no longer function. And also, if the conditions get very extreme, either end of the pH spectrum, you can have actually proteins denaturing. But what's going to happen before you get to these kinds of situations are the impacts of all these indirect effects of changing the pH. And lowering the pH affects the availability of some metals in soil, particularly aluminum. Aluminum is the biggest player. At low pH, suddenly a lot of the aluminum becomes mobile and available in soil, and therefore is can do its deed, can be toxic to microbes. Where if the pH was higher, they would just be all nicely tied up in mineral forms and wouldn't have any impact on the microbes. Also, the pH can affect things that aren't toxic, can affect substrates, things that microorganisms use, and so either increase or lower the availability of those compounds and therefore have some kind of effect on the microbes. Also, speciated organic compounds, things like that are speciated are going to also be pH dependent, and they will either carry a net charge or become uncharged because the proton will be taken up. For example, here it would become a carboxyl, and in general, uncharged molecules pass into cells more readily than ones in charged form. So that's going to affect also the impact of those molecules, be they nutrients, be they toxins on microbes. And this box here reminds you, if you don't already know, that microbes have an enormous effect on the pH of soil through their processes. So they are affected by pH, but they also affect the pH. And examples are when there's a lot of reduction in like a flooded soil system, and you're going to measure this in the lab when you do the flooded soil lab. You're going to see the pH rises as you go through these different electron acceptors, and protons are consumed. There's a net consumption of protons under those conditions. Oxidation reactions, for example, oxidation of sulfur and ammonium, and any sort of organic compound that produces CO2 is going to release protons and is going to lead to some acidification of that environment. And fermentation is another microbial reaction that produces organic acids as one kind of byproduct of those reactions. So if you look at the relationship of different groups of organisms and what range in pH they tolerate and what's sort of their optimum pH, there are some patterns here. These are not, these generalizations don't apply to all organisms in these groups, but in general I think that we can make these kinds of statements. Bacteria actually rule across a broad range of pH, although most of them are going to fare best at around neutral pH, 7 being a good value, but actually some of the most acid-tolerant organisms are not fungi, which you might think is true because fungi do a good job at low pH. They're actually bacteria that you see in acid mine drainage, where you can see bacteria at pH 0. I mean, at these impossible possible conditions, and they might be, archaea actually as well. Actinomycetes tend to not do well at low pH, and they are more likely to be found in alkaline conditions, and again may have an optimum of around 7, but the range that you're going to see them active in is 6 to 9 or 10. Fungi are tolerant of low pH. They may actually prefer a higher pH, but they can function pretty well at lower pHs in soil. And actually we sometimes use acidity to shut out other organisms, like to sort of reduce the growth of bacteria and make room for the slower growing fungi to take hold on plates. Cyanobacteria also are like actinomycetes and liking higher pH. You don't see them very much under acidic conditions. And protozoa as well aren't going to go to the extremes as much. They're going to be more in this range of 5 to 8. Again, I feel a little bad putting these numbers in this table, but at least you can get an idea of some of the general patterns that you see. What's interesting about hot springs Oh, that's temperature. Okay, skip that. What I was going to say is, in hot springs, you have these extreme environments, and they might be also associated with pH extremes, but it's probably more likely temperature, where the protozoa are shut out under the more extreme conditions, and there you see these enormous, like massive outgrowths of bacteria and archaea in those situations, because there's no predators there to be like knocking them back and preventing them from really taking over and growing these enormous mats of organisms. All right, and we use this terminology acidophiles, acid lovers, and alkalophiles to describe organisms that do well at low or high pH. Temperature. So temperature is probably one of the more predictable environmental factors, where we can have a better understanding of the relationship between microbial rates and temperature that is easier to model than is the case for other environmental factors. So this picture here is a really important one. It shows you temperature, and it shows you 24-hour cycle in soil. And if you look at these different curves, they represent different depths in soil. So you can see that the most extreme changes in temperature are at the surface of the soil, and you'll still see these variations as you go down deeper into soil a bit. But at some point, you're going to hit a depth of soil where it says, I don't care what's happening in terms of the diurnal pattern outside, on the surface. The temperature is dampened at these lower pHs, and you see very little response to changes in temperature over the day. So right there you can see that organisms in the surface are going to have to be much more adapted to these extremes in temperature, while those deeper in the soil have sort of a more predictable life they're leading in terms of temperature. And how hot a soil gets or how cold it gets is going to be a function of the color of the soil, with darker soil absorbing more heat, cover over the soil, whether there's plants growing on it or mulch. It's going to have a big impact on the exchange of heat across the system, and also water content. A wetter soil is also going to have a higher specific heat, and it will take longer for that soil to heat up and to lose its heat over time. So the relationship of temperature to microbial activity is shown by in this graph here, which shows you growth rate. Could be a process rate too, some sort of metabolic reaction, and temperature on the x-axis. And there's some minimum value at which there's no activity at all. It's very cold, and at that point you're looking at gelling of the membrane, and things just aren't going to transport across the cell, so there's not going to be much activity. And then as you increase the temperature from this minimum, there's this phase that this range at which you're going to have a pretty much linear relationship between the reaction rate and the temperature. And this is this predictable range where you can make pretty good predictions of how the rate will change as the temperature changes, which is really interesting for climate change. And then it's going to reach some optimum rate and then fall off from there, where at that point the temperature leads to denaturation of proteins and collapse of the cytoplasmic membrane, perhaps lysis of the cell. And this region in here, where you have this linear relationship, is could be one way to Q10 relationship. So Q10 is an expression that is looking at the, sorry, I'm not ready to describe this. So Q10 equals the rate, which is a K, at a particular temperature, divided by that rate at a temperature that's 10 degrees centigrade less than that first temperature. So you're looking at this ratio of the rate at a higher temperature to that same rate at a lower temperature. That Q10 value, that ratio, rule of thumb, is about 2. You might see it ranging from 1.5 to 3. So what this means is the rate will double with every 10 degrees centigrade increase in temperature. A nice little rule of thumb. And you can use this reorganization of the equation to predict what the rates will be from like based on knowing the behavior at one temperature and knowing what the Q10 is. And archaea are really good at high temperatures because they have these membranes that are much more stable at high temperatures. Okay, and then this picture over here shows you just some names of organisms. We humans like to give things names, so this is thermophiles, mesophiles, and psychrophiles, which are names for organisms that live perhaps obligately at higher temperatures, medium temperatures, and lower temperatures. And you can have organisms that can function up to 100 degrees and even higher centigrade. So there are organisms that can tolerate really tremendous heat, and some of those would probably be archaea. And let's see, I think that's, oh, and so what I wanted to say is, as the planet is warming, there's been a lot of research done on trying to estimate what's going to happen to, for example, respiration rates, microbial respiration in, say, the Arctic or in tundra environments. If the temperature increases by some amount, how much higher are these rates of formation of CO2 going to be? So there's been a lot of people who have measured these Q10s, like in laboratory microcosms, or sometimes in larger field scales studies where they can manipulate temperature. And they're finding that this Q10 is sometimes quite a bit higher than this rule of thumb of 2 as well. There's been a lot of interesting discoveries of different kinds of Q10s for particular kinds of microbial reactions. So, as usual, nothing is as simple as I'm telling you, but it's still a very good rule of thumb. And people use this relationship in models that are predicting rates of biodegradation or rates of nitrogen release from compost and organic farming or different kinds of microbial processes. Okay, water. Water is really crazy. Water interacts with almost every other soil factor. And for example, if you increase soil water, and you must remember this from the part 1 of this lecture, you increase soil water and you're decreasing the gas phase. So what happens is the diffusion of solutes or whatever, or could be gases, in soil are going to be really strongly affected by this ratio of water to atmosphere. And diffusion, rates of diffusion of, for example, oxygen are 10,000 times slower in water than air. So that ability of the atmosphere to restock these microsites in soil where there's carbon and all the oxygen has been used up locally is going to be much slower to replace that oxygen that was used if you have a lot of water in the soil because of these slower diffusion rates. So also, as you change water, you're going to buffer temperature more because of the specific heat of the soil will change, and also the osmotic potential that, for example, if you had a really salty soil to start with, when you put when more water enters the soil, you're going to dilute those salts, so the osmotic potential will go closer to zero. Also, increasing water can alter pH. Again, diluting the protons, it can increase mass flow of solutes through the soil pores and increase the diffusion of nutrients. And we looked at that before. You remember when we were looking at a soil aggregate and thinking about the path that a nutrient has to take to get from one spot to another. So it's kind of hard to put good numbers on where things drop off, but there's a few rules of thumb, I think, that we can come up with. And this figure is really awful, and I don't know why it looks this way. But anyway, this is megapascals here in this figure, and we have here, this is, I'm sorry, these numbers, this is wilting point here, minus 1.5 megapascals. And here's field capacity. So you still have, let's just focus on the wilting point, where plants are pretty much not going to be able to obtain water from the soil anymore. It's going to be held so tightly. You're still going to have a lot of microbial activity. Even as the water potential gets more and more negative, going to like minus 10 megapascals, you still will have some activity of fungi. You may have some bacteria dropping off, but you still will have some fungal activity. At the wetter soils, we're going to see more protozoa and zoospores and generally more bacteria in general. And they're going to drop off, especially protozoa and zoospores, and these motile organisms are going to drop off at much lower moisture contents in soil. So the numbers don't matter as much as just the these general relationships. Okay, and then what's probably most significant in soil are wet-dry cycles, is when the soil goes from being very dry to suddenly there being a rainfall event and the soil is flooded with water all of a sudden, and you get big shifts, and some of it are these osmotic shifts in soil. And this will affect organisms as well, and gram-positive generally survive those shifts better than gram-negative organisms. So it isn't just the water content, but it's also the changes in the water content that are important. All right, and here's some densities of different groups of organisms. These are just plate counts, but you can see here is this is PF. It's another measurement of soil moisture, but all you need to know is this is a very wet soil over here and a very dry soil over here. And you can see in very wet soils, your numbers, your plate counts are lower. And then as the soil goes into towards field capacity and all, you're going to get these increases in all groups of organisms. But then as the soil starts to get drier and drier, you see the bacteria drop off and the actinomycetes, and the fungi are actually not dropping off as precipitously from their peak as are, for example, the bacteria. And this picture here shows you respiration, relative respiration. This is like 100% respiration, and this is a bunch of different soils, but all showing the same general pattern as you move from, in this case, a dry soil to a wet soil, you see lower rates when it's very dry, then it reaches some maximum, and then it's going to fall off again when it gets wet. And these are looking at the gross activity, like a net activity of everybody present. You definitely see these kinds of patterns, even though you might actually go, if you looked at particular groups of organisms, you may find that they have some different relationships with respect to water. But if you add them all up together, this is a very typical kind of pattern that you see. Okay, osmotic potential, we just talked about it. Turgor is maintained in cells by there being a higher solute concentration inside than outside the cell. And you know this from osmosis theory, that if you have like a bunch of happy cells and you suddenly put them in distilled water, this is an extreme pure water, then the cells are going to swell up and burst open. And this could happen with a very rapid rainfall event, I suppose, in soil that was salty to start with. And in contrast, if you put cells in a concentrated salt solution, for example, in a very arid soil, then the cells are going to shrink and shrivel up through this process of osmosis. So what do organisms do to try to deal with this? They may actively pump salts inside themselves or outside themselves, depending on what they're trying to deal with, to reduce this gradient in solute concentration across the cell. They may also manufacture intracellular chemicals. They might be organic compounds like proline or glutamate that will help regulate osmotic activity. And then some have very rigid cell walls, for example, the gram-positives, and some of the halophilic salt-loving organisms have rigid cell walls that just make them more resistant to the impacts of salts. And archaea, again, because of their special membranes that are very stable in the presence of different tough conditions, they also do well in high salt concentrations. Toxins. So there's numerous kinds of things that might be toxic in soil, and I think this is important down here that, I don't know if this expression makes sense, and I always get these expressions wrong, but one dog's meat is another dog's poison. I don't think that's how it goes. But anyway, you can't really make generalizations about all microbes, because what could be toxic to one group of organisms isn't impacting another group. You know, an organic compound that could be toxic to one group could actually be a really nice carbon and energy source for another group. And there's, you know, some organisms that carry resistant genes that help them deal with the presence of toxic chemicals, for example, metal-resistant genes. So with toxicity, we're often talking, until we get into really extreme conditions, about different responses of different groups of organisms. But some of the kinds of toxins that you might find in soil that might impact microbes are heavy metals. Around like smelters, there's been some nice studies showing like drops in microbial biomass in soil immediately adjacent to these smelters that might be processing metals. And then as you move further and further away from the smelters, the microbial biomass is going to go back up and then reach a background level. Also close to highways, there's fallout of metals from automobiles, from tires, from road wear, that also can reduce microbial biomass in soil close to the roads. There may be antibiotics in soil. We know that there's a lot of organisms that make antibiotics, and it's kind of hard to demonstrate how they're actually working in nature, but we're pretty sure that they're used by one group of organisms against perhaps another in competition. Organic acids, so some just like byproducts of microbial activity may be toxic sometimes to the organisms that carry out the processes. I've already talked a little bit about suicidal organisms. They don't necessarily know how not to harm themselves. And also CO2. If CO2 can't diffuse away when it's made and it stays sort of localized and gets to a pretty high concentration, it can also be toxic to microbes. And then there's all these wonderful things that we humans have made, these strange new molecules in pesticides and industrial pollutants, some of which are toxic to some microbes, and others, as I said, will be used by them as substrates, and that's what biodegradation is using those. So soil organic matter and or clay, remember we talked about cation exchange capacity. They can reduce the toxicity of these toxins if they have the right kind of chemistry, by reducing the bioavailability, by reducing the access of those chemicals to microbes. And for example, if the if the toxin is a heavy metal like positively charged, if it sticks to clay, it may be present at a high concentration in the soil, but the microbe can't see it because it's held tightly to the clay and just simply is not there to be taken up. So it's not really toxic. And also hydrophobic compounds, compounds that hate water and will partition into organic matter. This will also affect their bioavailability and reduce their toxicity. Or if the substrate is a nutrient, may reduce access of that to organisms. It can work either way. So the assumption is that when we're looking at compounds that microbes use or that are toxic to microbes, only what is in solution in the water phase is what can be metabolized or taken up by microbes. So some of the processes that are involved that reduce the concentration in solution, because part of the a fraction of that solute, that substrate that we're talking about, is going to be associated with soil surfaces, clays, and organic matter through sorption. We already mentioned this before, I think in the same lecture, but sorption is a process where the substrate is held at the surface of particles through different kinds of chemical associations. Also, some substrates are not very soluble in soil, and they will precipitate out of solution or just be created that way. Like a lot of plant materials are very insoluble, like cellulose and lignin, and therefore their availability to microbes is very low, and often we need other organisms to help break them into smaller pieces that go in solution and then can be taken up. And also remember, we just talked about speciation. Depending on the pH, some compounds are charged or uncharged depending on the pH, and that affects their availability. And I think what's interesting is to think about sort of scale, and if we're thinking about like a pesticide that has been applied to soil and has the potential to be biodegraded, then if it in the soil, like the organic matter may tie up this pesticide through sorption and lower the potential rate of biodegradation of that substrate. And so in the short term, it might end up being a slower rate of removal of that pesticide. But on the other hand, the fact that the pesticide is hanging around in that surface soil, remember we looked at the changes of in density of microorganisms as we move deeper into soil. This is really sharp drop off from the surface into the, as you go deeper into the subsurface. That means that if that pesticide hangs around in that surface layer of soil, it's ultimately going to have some potential to be biodegraded, because microbes will probably finally be able to get their hands on the substrate. But if the pesticide did not linger in that surface area and hang around until microbes can utilize them, then they'll just blow through the surface and move on down to the subsurface, where there's going to be not much potential for them to degrade, and it's likely that the pesticide moves into groundwater. So there you have this funny kind of contradiction. As a function of scale, sorption can decrease the rate of biodegradation, but ultimately may permit conditions that would help the pesticide to degrade over a longer time frame. I hope that makes sense too. Okay, and then just a couple of terms here: compensation and synergism. So you never have just one kind of factor playing out at a time, or you rarely do. And so you can have these compensation reactions where one factor may alleviate the effect of another factor. And an example is like some archaea that could not grow at less than 0 degrees centigrade because it would be frozen. However, in the presence of high salt concentrations, it can degrade. So the salt is compensating for the effect of the temperature. Where you might predict it, like, oh, no way that can be active at less than 0 degrees. However, if that soil is very salty, it could be active. And synergism, where one factor enhances the effect of another factor. So the opposite. In this case, like nitrifiers are going to be more sensitive to a low temperature if they are also in a low pH environment. So again, when you're trying to like create these mathematical models to predict rates of microbial activity, you may have to be concerned about these potential interactions of different factors. Another really important, interesting thing about soil, again, has to do with scale. A lot of microbial processes that we're going to measure in soil are measured at sort of a bulk scale. That means we're not measuring it at the scale that the bacterium, for example, is living at. It's not going to be a measurement at like a 1 micron scale usually. Instead, we're going to be lumping together many different microsites that are containing different sorts of physical, chemical habitats, microenvironments, and also different associations of microbes. And that means that we can really miss out on what's going on, because we may, for example, be lumping together aerobic and anaerobic microenvironments and making a measurement on it and getting some in-between sort of measurement of a rate, when actually we're perhaps looking at two very different rates that are occurring in these two different microsites that we've lumped together. All right, and then let's talk a moment about sterilizing soil. So sterilizing soil is really important in studying a lot of biological processes in soil, particularly if you're trying to tease out what is the role of a biological process. How much is it actually, for example, the microbes involved in the process versus just physical phenomena that are going on in the soil. And I think, again, it's good to think about biodegradation as an example for this. In environmental science, we want to know, will this pesticide that's used in soil biodegrade? And so you take it in the lab and you try to study it. And if you just put it in a flask with some soil and you see it go away, you might say, oh, good, it's biodegraded, no problem. Make some prediction of how fast it takes to degrade. But actually what you might be looking at is not a biological process, but the chemical might be just volatilizing, or it might be sorbing to the organic matter and still be there, but your extraction method can't pick up the sorbed phase. Or it could be a chemical reaction that's going on, and it has nothing to do with biology. So if you sterilize soil, you can then compare the non-sterile biologically active soil to the sterile soil. And some of the ways that we sterilize soil, and none of them are perfect, all of them are bad because they affect the soil in different ways. But here's what we have. There's can be sterilants that completely sterilize the soil, and there can be inhibitors that just knock back activity in soil. And you see both of these in studies like this. Mercuric chloride is a sterilant. So one approach is to put these chemicals into soil and knock out any sort of biological activity. Sodium azide is another example, although sodium azide loses its effectiveness pretty fast. Mercuric chloride is with you forever, and plus you have to pay a huge amount of money to dispose of it through environmental health and safety. So don't use mercuric chloride. You're just creating an environmental problem by studying an environmental problem. You know, if you're studying like biodegradation and you use mercuric chloride in your study, it's crazy. You're creating a new problem. Okay, autoclaving is another way, and often autoclaving involves 2 to 3 autoclaves, separate autoclave events with incubation periods in between. So like you go put it in the autoclave and clobber it, and then you put it at room temperature, and you encourage all those endospore forming organisms, those Clostridia and Bacillus, to like who managed to survive the first autoclave, to like come back out and take over. Everyone else is dead, and plus there's nothing like another microbe is the best, you know, carbon and energy source. It's nothing like eating your brothers and sisters. So then they'll bloom and grow up really to high populations in the so-called sterile soil, and then you hit them again with another autoclaving blast, and then incubate them again, and then hit them again. So that's a traditional way to sterilize soil. But I have to say that by the time you've autoclaved the soil that much, even once, you're changing the physical and chemical properties of the soil quite a bit. And that might not be acceptable in your particular kind of experiment if all you want to do when you sterilize the soil is just remove or knock out the organisms. Then you're going to be in trouble, because you're going to have a very different physically soil than before you autoclaved it. Heat is used sometimes. It's not very effective. It can knock out some of the life in soil, particularly the soil fauna, and maybe the fungi, but it's actually a process that a lot of people have access to, because a lot of people have ovens, but it's not used very commonly in research. The gold standard is gamma irradiation. It's to expose the soil to a source of radiation. There are facilities around. I think there's one at maybe not UC Davis, but a facility associated with UC Davis that is used in the medical world to like sterilize, I don't know, organs or something that they stick in, they replace your bad organisms with, and they need to be sterile before they go in. But anyway, you can often go and set up your soil to be autoclaved at these sources. So that's supposedly has the least impact on the physical chemical properties of the soil, and is quite effective at removing organisms. Okay, and then I think on Friday we're going to do a class exercise where we're going to review the information that was presented in this lecture, So I'm just going to stop right here, say good night, sweet dreams, and see you next time.